This is a revision video on diffusion. So there are 8 revision questions in total throughout the video. We'll start with 3 on simple diffusion. So pause, try the questions, and then we'll go through answers for these 3. Okay, so your definition of simple diffusion. The key thing here is from a high concentration to low concentration, down a diffusion gradient. For 2, let's have a look at the molecules then. So we've got oxygen, that can diffuse by simple diffusion. Glucose can't. Carbon dioxide can. Amino acids cannot. Sodium ions cannot, and other ions can't as well. Fatty acids can, and ethanol can. So I'm providing the structure of ethanol there just to help if you don't know it. So the key thing here is this. The molecules that can diffuse by simple diffusion through the membrane are very small and are non-polar. There are some cases where the molecules are large, such as fatty acids and ethanol, but they are lipophilic. They are non-polar and can move through. But in the case of ethanol, it does have this polar OH group, but it can still dissolve lipids. It's still soluble. And you'll remember that it's used in the lipid emulsion test to dissolve lipids. So question 3, still with water. So it can move through by simple diffusion, but it also can use protein, specifically called an aquaporin. And so water can cross the plasma membrane by simple diffusion and facilitated diffusion. Okay, so now let's look at facilitated diffusion. 2 questions. So pause and answer those. So the differences, well, facilitated diffusion uses carrier proteins or channel proteins. Remember, the key thing is those proteins are specific for the molecule they transport. So they will only transport one specific molecule, or they might transport 2 at once. But they are specific only for that molecule. They won't carry other molecules. And their similarities, both a case of diffusion. So down a concentration gradient, high to low concentration. And if it helps, I can just show you here. So we've got a carrier protein in this case. We've got molecules at high concentration at one side of the membrane, at low concentration on the other. The molecule will be specific, see how it fits into that binding site. That causes a change in shape of the molecule, and so it can move into the other side of the membrane. And then because it's moved out of the binding site, the protein changes shape back again so it can transport another molecule. In the case of a carrier protein, it's almost like a tube through the membrane, but it still is specific for only one type of molecule. So in this case, let's say they're ions, they can just diffuse through that channel protein. So 2 questions here relating to the graph. Pause, try those questions, and then we'll go through the answers. So for question 6, you should have this. A and B show simple diffusion, C is facilitated diffusion. Why is that the case? Well, if a substance is diffusing by facilitated diffusion, then it requires carrier proteins or channel proteins to move through. And obviously, the number of those carrier proteins or channel proteins in the membrane limits the rate of diffusion. So you can see line C plateaus. The carrier proteins or channel proteins in that case are saturated. So there's your answer. So question 7, we know A and B are the lines for simple diffusion, so it's one or the other. So this should be your answer. A is a cell with microvilli, B is a cell without. And the reason for that is that A is occurring at a faster rate. And we know that microvilli increase the surface area of the cell membrane. And so if you've got a larger surface area, you can increase the rate of diffusion. So question 8 relates to the graphs below. So in each of them, membrane permeability is on the y-axis, and there's a different independent variable for each on the x-axis. So you have to just plot a line for each. So pause and do that, and then we'll go through the answer. So for pH, as we go from an acidic pH to a neutral pH, the permeability will reduce. And if we were to put a cell in alkaline conditions, with increasing alkalinity, the permeability will increase. For ethanol, going from a 0 concentration up to 100%, there is a gradual increase in membrane permeability. And finally, with temperature, the graph looks pretty much the same as with ethanol. The reason for these observations are given here. So in the case of pH, those extremes will denature membrane proteins. Obviously, some of those are going to be carrier proteins and channel proteins. So if they're denatured, they no longer can regulate what passes through. And as a consequence, permeability of the membrane increases in either acidic conditions or alkaline conditions. Neutral pH, around pH 7 is going to be optimum for the cell. Again, for the case of temperature, we know that increasing temperature, increased kinetic energy, and denaturation of proteins at high temperature. And again, it's the same effect. You denature those carrier proteins and channel proteins, and they can no longer regulate what moves in and out, so the permeability of the membrane increases. And with ethanol, you should remember that ethanol is lipophilic. It can dissolve phospholipids, dissolve lipids, and so as you increase the concentration, you dissolve that phospholipid bilayer in the ethanol, and obviously then it can't act as a barrier, and the permeability increases.